We're here at the 17th CROI, and we're here with a group talking about prevention, some PrEP, and what is jumping out at them at the conference. So we'll begin with Maren Cohen. We'll introduce as you present your material.
I'm a professor at the University of North Carolina at Chapel Hill, epidemiology, microbiology, and medicine, and I'm one of the co-directors of the HIV Prevention Trials Network. I think that my other esteemed colleagues will talk about current activities. I'll just say just a word about what's in the future, what's coming down the road. We're doing trials now of long-acting injectable agents, and we're hoping that both men and women could receive a single agent through an injection that would last about 6-8 weeks, if we're lucky, around 8 weeks. And that drug we're testing right now is called cabotegravir. It's an integrase inhibitor. We're about 3 years away, roughly 3 or 4 years away from knowing how well it works. My colleague Deborah will talk a little about the trial designs of how we look at this. Another thing we're very interested in is monoclonal antibodies. That's a whole other way of preventing HIV. We're doing a trial called the Antibody-Mediated Prevention Trial in collaboration with the Vaccine Trials Network. The antibody we're using is called VRC01 that blocks the binding site, the CD4 binding site. That trial is well out of the gate with almost 1,000 people, more than 1,000 people enrolled already. And we're hoping to prove that antibodies work to prevent HIV acquisition. There's a lot of other antibodies in development that could be used as subcutaneous antibodies instead of an infusible agent. We're infusing this antibody every 8 weeks. The newer agents that are going to be available to us last longer and could be given subcutaneously. So we're very excited about really developing antibodies for prevention concomitant with these long-acting antivirals. And lastly, we're working on helping investigators develop implants. There's several different kinds of implants that could be used, like birth control implants, that might allow a person to have an implant that would last months or a year even, with an antiviral distributed for prevention. So those are the things we're working on, and we're quite excited about it, and it's making good progress, and hopefully we'll have some new products.
Good. We'll talk to your colleague here.
Deborah.
Yeah. Hi, I'm a biostatistician, and I have been working on HIV prevention trials for a goodly number of years. And I think there's one interesting new aspect of the trials that are being done right at the moment. So Mike mentioned we're doing some trials of antiretroviral agents, and we're also doing some trials of monoclonal antibodies, and they are different mechanisms. And one of the things you'll find out about those different trials is that if you are entering a trial that's actually using an antiretroviral, so cabotegravir, a long-acting injectable, is an antiretroviral drug. In those trials, we're actually directly comparing an antiretroviral, cabotegravir, with another antiretroviral, which is proven to work, Truvada. So when you enter those trials, irrespective of which product you get randomized to, you will be receiving an active antiretroviral for prevention. However, what's different about the monoclonal antibodies is we don't currently have human data about whether monoclonal antibodies actually reduce HIV infection. So if you were entering a monoclonal antibody trial, you will be randomized to either get an active infusion or a placebo product. At the same time, in the background, you have a choice about whether or not you take Truvada. So I really just wanted to point out that those, while it may be a little bit confusing, that in one trial you always get an active product, and in the other trial you're randomized to a placebo versus an active monoclonal antibody. The reason for the difference is really that in one case we know antiretrovirals are very likely to reduce infection, whereas in the monoclonal antibodies we've yet to establish that. So I just wanted to talk a little bit about that confusing aspect of why you get different things in different trials.
But depending upon the results, you might have a DSMB stop the trial prematurely, or
That is true. In any of these trials, these randomized trials, we keep a very close eye. There's a group of independent experts who review the trial about every 6 months. And if there's compelling evidence that we know that it works, we stop the trial straight away and let the world know what's going on.
There's always those safety precautions.
Yes, there are always those safety precautions built in.
And Jared?
So CROI is always such a great conference because it's full of stuff, and I'm trying to think of the things that make me most excited, and there are probably two big ones in our short time that we've got here. The first is just how normative discussions about PrEP have become, and how it's really in every prevention session, now including some of the surveillance sessions that were this morning to say there's a high incidence community, what's the plan for PrEP there? Comes up in the questions. And as a result, a lot of the research being presented here is on innovative strategies for delivery. In fact, this next session that's starting right now is about different ways to deliver PrEP, including through pharmacies, where you don't have to see a doctor at all, including using text messages and other strategies to improve adherence, and other great ways to bring PrEP out to people in real-world settings, because it's not as complicated as it might, as you would have thought at first. You still have to do testing, but make it simple so that it's accessible and so that it has impact. The other stuff that's exciting for prevention here this time, beyond what Mike and Deborah said, which is looking a few years ahead, is sort of the next steps in the stuff that we already have. So PrEP and its interaction with biology. So there's some nice abstracts tomorrow about PrEP in women and its both topical and oral pills like Truvada, and its interaction with the vaginal microbiology, and what might help PrEP work better and might help some kinds of PrEP work less, as well as the safety of dapivirine ring, which of course was presented last year here at CROI. And this is more information being presented tomorrow and Thursday on safety in its interface with contraception and in women who happen to acquire HIV in spite of its use. Kind of interfacing a lot with kind of things Mike said about things for prevention that prevent pregnancy and HIV at the same time. If we can pair together active PrEP agents as rings, as pills, as shots, as implants with active contraceptive agents, they might make it really safe and really desirable for populations. Really excited about all of that kind of multi-purpose discussion that's going on.
Great. Last one. You have to fill in the last of the gaps.
Okay. Well, I would say that I'm an ID physician in Paris, so in Europe. In Europe, we are also interested in PrEP, and we are rolling out PrEP now in a number of countries where PrEP has been approved. We are, continuing our research with the support of the NRS on trying to assess new dosing regimen for PrEP. We're using in Europe, apart from daily PrEP, on-demand PrEP at the time of sexual exposure, and we have now, more and more data to support that this dosing regimen also provides full protection against HIV acquisition. The other topic we're interested on, and we are presenting some data at this conference, is actually addressing.
Excuse me, I think your mic just fell.
You lost your microphone. Not that it wasn't interesting.
I was too enthusiastic about what we're doing in Europe.
Start over.
So the other topic of interest for us is the issue of STIs, sexually transmitted infections, because we've seen among high-risk individuals on PrEP pretty high rates of STI. It's true also that over the last couple of years, we've seen in a number of countries an increased rate of STI, and that predated the implementation of PrEP. But I think having all these PrEP programs implemented in a number of countries now provides a unique opportunity to do more research in STI and more research to do a better job in terms of how to test for STI, how to treat STIs, and how to prevent STIs. And we are presenting new data at this conference, trying to assess new strategies to reduce the incidence of STI among PrEP users.
Maybe it's appropriate for me to ask, because you are the thinkers in this field, and I thought it might be good to ask you, is there something that you see a void that needs to be filled with work, with research that isn't being filled properly?
Can I add one thing? One thing I think is interesting with the STI problem, I'll call it. Maybe it's not surprising that people on PrEP are using condoms perhaps less efficiently or less frequently, and STI incidence is increasing. But what I think was a fear of people who study transmission, the biology of transmission, was that the inflammation of the STI would overwhelm the PrEP. Okay, this was not more than a theoretical fear. We were very concerned about it. What's happened to my knowledge in the clinical trials is we've just not observed that if you take Truvada, that barrier seems to be sufficient to block the amplified transmission that would normally be observed with an STD. So I think that's a piece of good news about Truvada. However, I think that for those developing new PrEP like us, we have to be concerned that every drug has to reprove itself. For example, in the integrase inhibitor cabotegravir, that may or may not be able to withstand the amplified acquisition inspired by an STD. So I think that's an important consideration for PrEP developers, actually, the STD problem. Because it's not just another disease, it also amplifies the acquisition of HIV. It makes it easier to get HIV in the face of innocence.
Well, I think it would be fair also to say that, we haven't seen such an increase of STIs in people using PrEP. It's just that these people are at high risk already of acquiring HIV and STIs, and now with Truvada and other drugs in development, we have shown that, despite the high rate of STI, you could fully protect from HIV acquisition. But I think, like in other areas of medicine with HIV research, we have been able to also foster research in other areas, in hepatitis, and now I think the field is open also to do more research on sexually transmitted infections. And we will leverage probably on the studies we're doing on prevention of HIV to also address the issue of what could we do better in terms of prevention of the other STIs in the future.
We have one more thought here.
Yeah, I think just in terms of your question about what is the gap, I think what we have realized in the last 5 years now with PrEP is that first with Truvada and then with maybe the ring and then with with injectables coming up, is that every one of these drugs has to prove itself at its biology and then its ability to how it's going to be delivered and who it can be safe for and how does it work in the context of STIs and contraception and pregnancy and everything else and then all the way through to how to get it to people. And just because you make something fancy doesn't mean we're going to get it to people and make impact, and we need to be pushing the research all the way along and pushing the things that are a little bit further forward so we can be having impact now. It's still as much as we talk about PrEP and prevention, there are still many places, including everywhere where we live, and then everywhere where we don't live, where there's not enough use to be having as much impact as it could. And new options that provide choices to people, like a ring, like an injectable, like an implant in the future, are going to need the whole push to be able to have the impact later.
Yeah, I mean, I would say if I was thinking about the gaps, that the one thing that still puzzles and confounds me a little bit is the issue about the adherence issues we have seen to products in women. Like, so both in the ring and in the Truvada trials that we did, for reasons that we haven't managed to overcome, women were not very adherent to those modalities, and they're not going to work unless people are willing to use them, and what we have to do to make them more appealing or to understand why or to understand the stigma, I think that's still a gap that is a little, it's still an unknown problem, exactly why people were not willing to use these products.
Thank you very much. I appreciate it. You all have done a great job in a short time, packed in there, and it got the point across, I think. Thank you very much.
Thank you.
Welcome.